Hello, welcome back to COG 376, Computer Models of Language Representation and Processing. Last time we talked, no, that's not this at all. Hello, welcome back to COG 266, Brains, Minds, and Consciousness. So, last time we talked a lot about the, hang on, adjusting the microphone, about all the different ways through history, perhaps not all, by the way, but neuroimaging techniques. How do we get an image of the brain and the things that the brain does? Imaging versus staining versus all the ways that we can observe and measure the brain. So today, we're going to talk about some of the features of these techniques and things you might need to know about just how all these techniques relate to each other. So once again, we walked through sort of the path of history that brain measurement techniques have taken. We saw the stain transfers. We saw a little bit of electron microscopes showing us some 3D structure of neurons for once. X-rays, PET scans, CAT scans, all these wonderful things, up through brainbows, the strange genetic techniques of visualizing brain and neurons individually in the brain. So we see this progression a little bit. These all do slightly different things. It's not just a straight linear progression, but we see as the technology shifts, we see more options opening up. And the key thing that we develop is a shift away from only focusing on the structure of the brain and into measuring the function. So this is a really important distinction, and it can be a little fuzzy, actually, because obviously the structure, it must relate and determine to the function, right? If my brain was made out of bees instead of neurons, that would be quite a different brain, right? But the idea is that it's all about what we're able to measure, remember, right? So the focus is on measurement. So measuring shapes and contours of structures is a little different technology than identifying a process that is actually taking place within those structures. So because processes have to be identified as a change in some property over time, that's the only way you can really capture an ongoing process, right? It's extended in time. So consider the heart, the friend and compatriot of the brain. Hearts have structure. They also have a function. What's the function of the heart? Hey, move that blood, right? It actually is a little bit more fine-grained than that, but that's the general idea. The structure provides the tools for actually achieving the function. So we can talk about the heart in terms of its structure, which, break it down, we have the aorta, vena cava, all the different ventricles, right? All the different chambers of the heart, the different muscles that can move and help move the blood around, right? But the function that you measure in a heart is things like, for blood, that you might be tracking the amount of blood moving through the heart at any time. This is a change in time, right? How much blood flows through the heart. Same with pressure. Your heart is also responsible for keeping up your blood pressure. If there's a leaky valve, you might not have as high blood pressure as you need to do normal things. Or if you have some cholesterol issues, you might have way too high blood pressure. And these are functions. These are things that are measurable in time. Now, you might say, okay, well, pressure, you could definitely measure it all at once, but it's something that changes, right? And same with the function of the muscles in the heart, right? Are the valves all working? Are they all closing completely? Is the strength of the pump strong, or is it weakened for some reason? Is it arrhythmic, right? So that's the idea. You there is a structure, of course, to what we're trying to analyze and understand, but there's also function attached to that structure. So brain measurement began almost entirely with structure because, hey, that's the first thing you get access to when you start getting cool cameras, right? I can take one image and get a good idea of what the structure of the thing is. So when it comes to measuring the structure, you're trying to create a detailed image, but a static image, more or less, of the thing in question. So we started with X-rays, CAT scans, regular MRIs do this. They are trying to capture in as fine a detail as possible using magnetic fields where are all the neurons? Where are all of the large scale structures of the brain? And then we also saw the electron microscopes, right? The idea of being able to look at individual cells at this level of detail that was unprecedented. And the idea of measuring structure is you're more or less doing this all in one shot. You're just getting a snapshot of what is there, right? And trying to make it as detailed as possible. With the heart, you're targeting muscle tissues, right? What is the arrangement of the ventricles, right? And with the brain, we're aiming more at neural tissues. So heart, just to poke our head back in to that side of things, it has all these different tubes, right? You can think of yourself as a series of tubes, much like the internet. So you are an interconnected network of tubes, and the heart is where a lot of them meet. We can also look at, recapping the electron microscope image, right? This is incredibly detailed structure. We are seeing down to the little bulbs at the ends of the neurons. We're seeing the dendrites and the cell bodies of these things. It's incredible. It also takes a lot of energy and a lot of effort to get something that is this fine detailed in terms of being able to look at this, nanometer scale of the brain. Well, probably not nanometer, neurons or whatever. So CAT scans are also on the list of structural things. These are, remember, the slices, the X-ray slices of the brain, top to bottom. And, here we're actually getting a 3D image of the whole brain altogether. Now, it's not as detailed as the electron scan, but then again, it's as detailed as it needs to be to capture the level of complexity that we're trying to hit. We're trying to say, what does the whole brain look like, right? And remember, this kind of scans down from the top. Actually, I guess this one looks like it starts from the bottom in the upper left. So you get a lot of face bones. Hit the eyes, and then you go up through all the fun slices up through the head, right? So these are the things that we developed over time that measure structure. Measuring function is a different ballgame. So to measure function, you have to identify a change in some feature over time. And this is key because the idea that we're adding the dimension of time and not just space to what we're measuring creates this sort of tension. How often am I able to measure the thing? Because that helps me see things better in terms of the time scale. But also, how good is my image, which helps me see actual structures better, right? And on top of that, we're trying to target something very specific. So we saw magnetic resonance, radioactive glucose, and then electrical activity. These are some of the measures we can take. So with the heart, you're targeting the blood and its flow, but with the brain, you're targeting the oxygen or food used by neural cells, right? That we discussed how cells are hungry. Or you're getting things like the residual differentials in electrical activity. And again, it's great to be able to bounce X-rays off of physical structures and just say, hey, that's there, right? But to measure function, you have to really target a specific kind of thing. The bold response is a very specific thing that is available that we hope correlates with, more increased neural activity. There are other ways to measure these things. So back to the concept of the heart, the blood flow, right? It's not just the structure, it's the flow. Where are things flowing? What is the pressure? What is the movement? But what about EEG, right? So EEG techniques they have really good, they give us a good image of things extended in time. We'll talk in a minute about the idea of what's the resolution, right? But for now, just EEGs give us this ability to only look at a few nodes. So we're not capturing the whole brain in 3D. We're not capturing it even in 2D. We're capturing it sort of from this, I'm just going to arrange these nodes on your scalp. Here, I got 25 of them. I got 64 of them, whatever. And we're gonna ask questions about the readings that come off of those. So it's a really low resolution image, in a sense, marching left to right, but you're getting a lot of temporal accuracy with these techniques. PET scans, as we saw, they are measuring changes in tagged radioactive glucose, right? So you send in the sugars, and your brain starts to use them, and we're able to see them using this radioactive measurement tool. And again, that is the idea that we need to measure this over time. PET scans are fairly slow, similar to fMRI, which, if you recall, is the bold response, the idea of trying to measure when the oxygenation of blood changes in a particular area. So fMRI is giving us, we're still getting the structure, and we're getting some of it at least. But we are also getting a lot of the activity, right? Or what we hope correlates to a specific kind of activity in the brain, right? So this is the idea. We can be measuring just structure, or we can try to measure over time and get function as well. But we have to be very careful about what we're targeting. It's not just, hey, show me the neurons. Hey, show me the neurons again. It's, hey, show me some critical change in the neurons. Show me some important change in the food supply to the neurons, or the oxygen supply. So to talk about all of this, we need to talk about resolution. So measurement resolution. So the resolution of your measures is all about how fine-grained the details are of your particular measurement. In the images above, the far right is 100 times the resolution of the far left, which, if you look at it, is just 1 pixel. That's sort of the averaged color of the whole square, kind of smoothed into a goopy green, greenish, tealish something. Maybe blue, whatever. So it depends on how good your color vision is. So you see, I hope this illustrates very clearly, the image on the right is far higher resolution. At the same time, that's 10,000 pixels in that 100 by 100 block of pixels right there. If you think about that in just the basic energy cost, it's going to take you 10,000 times as long or as much energy to deal with that image than the one on the far left. So while structures are extended in space, they are just the physical structure of the entity that we're studying. That means that when it comes to measuring spatial resolution, it's similar to the above right there. You can have super fine grained details like an electron microscope, or you can have a little bit more calculating the average activity in a voxel for something like bold response in fMRI, right? Whereas function is extended in both time and space. So it involves this trade-off between spatial and temporal resolutions. Because if I only have one machine and it's capable of measuring either one thing 10,000 times or 10,000 things once, that's a trade-off. That's a balance. It's harder to get a lot of images in a row when you also want them to be more expensive, more detailed, right? So, spatial resolution. It's just the basic idea of how precise can we get with the physical, the sort of the spatial layout that we achieve. How many dimensions? That's one way to think of this. 2-dimensional image, 3-dimensional full models, or maybe just a series of individual readings all taken together in an EEG net. An EEG net gives terrible spatial resolution. It's only 64 different little single nodes, whereas, a CAT scan is giving about as fine detail as the film can get. So that's the other thing. What's the basic unit? What's the minimum distance between two things that I can actually detect a difference? So is it like X-rays on film, fairly arbitrarily small? Or is it averaged into voxels like an fMRI? This is the idea of spatial resolution. Every technique that you're using lives somewhere on this scale, on this spectrum. Temporal resolution is very similar. It's just you're thinking along a different axis. You're thinking about time. So what's the minimum time between two events happening that I can detect a difference? The faster the imaging, the higher the temporal resolution. So faster imaging, it's spending its energy on capturing more images, but with less clarity. EEG has terrible clarity when it comes to saying, here's a picture of the activity in your head, right? But it gets about 1 image per millisecond. That's amazing. That's 1,000 per second compared to fMRI, which gets, it has great voxels. It locates activity in 3D space in the head, which is fantastic, but it's 1,000 times slower than EEG. 1,000 times. So that means that to get these better 3D imaging techniques, these 3D images rather, of the activity in the brain, the bold response, that's a lot more energy intensive. It takes a lot more effort. Whereas EEG, those little resistance changes, the milliamp changes on the surface of your scalp, those can be measured pretty easily, pretty fast. And because we're only measuring 64 individual signals, hey, we good, right? So this is the idea of the trade-off between temporal and spatial resolution when we're trying to get functional images at least. Just a quick aside, structural imaging is always trying to focus on being as detailed as possible. So trying to maximize spatial resolution all the time with that. But remember, functions we have to consider them as a series of images over time, so it requires this trade-off. Good temporal responsiveness, sacrifice spatial resolution. Cover this again, repeating myself, but I think it's a key distinction that it really illustrates the point. EEG has low spatial resolution. It's barely 2-dimensional. It's sort of, here's 64 or 128 or 32, whatever. Usually a power of 2 for some reason. Just processing ability. Here's a bunch of wires touching your scalp. It's barely a map of your activity of your head. But we get 1,000 measures per second. So we can do things like track the way that your neural activity changes millisecond to millisecond. That's a powerful tool. Compared to fMRI, which again gives the full 3D head scan, right? I would call this moderately high spatial resolution because it's not nowhere close to our friends the electron microscope and things like that. It's these 3D voxels. They could be sliced up smaller if the technology was stronger, but it's pretty good. It's pretty solid. It at least locates things in 3D space within the brain, and it has moderately low temporal resolution. I'd say perhaps even heavily low, but we'll leave it in the moderate zone because it's kind of striking a balance. It only gets 1 measure per second, but that's better than, 1 per 10 minutes or 1 per hour or day or something, right? So it gives us this this scan of our brain, and we're able to see how average activity on fairly small voxels over 1 second. So the minimum time we can detect a change would be 1 second to 1 second. That works, but that also determines what we're able to analyze. Talk about more of that later. Here's another tool that we haven't talked about yet, magnetoencephalography, MEG. This is supposed to be the answer to, hey, you want that high level EEG type temporal resolution, but you want it on a full brain scan, eh? So spatial resolution is a few millimeters, which is pretty solid. That's comparable to fMRI. And temporal resolution that's similar to EEG. It's on the order of milliseconds. So this is a great tool. Now, there is one issue. It doesn't penetrate deep into the brain. So it might miss some activity from lower layers, deeper layers. It gets most of the top level of the cortex. We'll talk more about cortex in a future lecture about what the heck's going on up there. But the idea is that to get this powerful measure, we are still giving up a little bit. The resolution hasn't really gone down, but the effective range of what we're able to detect has gone down a bit. Still a powerful tool. It's not as widespread because it's very specialized and very specific, and every hospital in the world needs an MRI. Not every, but most. Whereas not everybody needs an MEG machine, so not as widespread. But let's just lay these all out together. So resolutions, on the x-axis we have the temporal resolution of a technique, and on the y-axis we have the spatial resolution. So EEG and MEG, they kind of stick to the top level of the brain and sort of cortical areas. MEG is much stronger. That's not represented here very well. But fMRI is able to go a little deeper in terms of the spatial resolution. But you see, it doesn't capture anything of anything less than a log of seconds. So 1 second. 1 second to longer, cool. You can capture all kinds of stuff beyond a single second, but nothing below it, right? Nothing to the left of 0 on this chart. PET scans are a little slower, take time, take hours. Lesions, I'm not even sure what we're getting at there. Damaging the brain for the sake of studying it. Fun. So this is the idea, though. I hope this image is helping to situate that there's always sort of a target area of what is measurable with your tool. Whatever imaging technique we are looking at. This kind of lives in this continuum. Well, this space. These two continuums. Continua? Okay, so okay, now we want to pull all this together. So we've established that different techniques are going to be sensitive in different ways. And there's always this trade-off between spatial resolution and temporal resolution and the scale of all this, right? So fMRI versus EEG, key. What we really want to do is combine the results of all these methods to get a complete picture of neural activity in various conditions and different perspectives, right? If I detect a certain kind of activity with fMRI, I should then be able to make predictions about what I might find with an EEG measure. They won't be measuring the same exact thing, but they will help us to sort of guide each other, right? They kind of bootstrap up and down the brain. So this is all because brains are hierarchically organized. We have, at the highest level, we think about them in terms of whole lobes, nuclei, different sort of specific areas that we know things about, down into, the architectonic areas and the connections between them, down to the neuron level, down to the synapses between, down to the molecular structures, right? So you never measure all of these at once. No tool is really going to do that. So the idea is that we need to ask questions about what's happening at different levels so that we can then make sense of more of what's happening in the brain. So again, different techniques are going to be sensitive to different levels of these organizations, of these different scales to show different functions. So local and long-range connections are going to serve different functions, but connectivity at the neuronal level is going to be different than large-scale connections, right? So a synapse is different than the big interconnected nature of, say, how your visual system sends things off to the spatial areas to make sense of the spatial organization of what you're looking at, right? So they're both about connectedness, but they're at different scales. Measuring what happens between just two individual neurons is important, but so is measuring what happens between huge populations of neurons and how they send data, send information forward. So the brain is multi-scale, so we need to work at multiple scales. So this is why I brought up resolution. The higher resolution, the finer the granularity, and it's going to allow us to detect more kinds of things. So higher spatial resolution helps us see all of the past the lobes, down into the gyri, down into the neurons, and so on, and perhaps even down into the chemicals. Whereas higher temporal resolutions get us different phenomena. The bold response, as we said, lets us see general activity within a voxel. And ERP, for example, one technique you can use in EEG, lets us pinpoint specific activity after something happens. So just a quick note on ERPs. ERPs are this thing you can do with EEG, electroencephalography. And the idea is that because we're measuring at the millisecond level, we can actually get, hey, I see this wave as it changes. So event related just means we are measuring after a known event happens. Say I make a bang sound while you're sitting in the EEG, and I am trying to measure what's your neural activity after that bang. So ERPs are that you do it a whole bunch of times and you average and you get this wave of, oh shoot, there's a dip, a refractory dip, and then there's a strong negative response. So negative is up in electrons, in neurons, because of electrical something. Don't worry too much about it. The idea is just that there is a distinct pattern here. We see going left to right that the measurement of the electrical activity in the head it dips down, spikes up, dips back down, and then there's sort of a refractory period, right? That's this particular ERP. They all have different shapes. They all kind of mean different things. That's a whole subfield you could dig into. But the idea is that without the temporal resolution of an EEG, this would just be a flat square. Because all an fMRI is able to measure, even though it's able to isolate down to very specific areas in the brain, it would only be able to measure one block of averaged activity over twice this span. I couldn't even fit it on the slide. Twice this time, right? The entire ERP event, the interesting stuff, all the little spikes and peaks and valleys, gets smoothed out. It cannot be measured by something that doesn't have sufficient temporal resolution. So that's why it's such a key concept to think about while we consider these elements. So now I want to show you a nice scale that helps to sort of situate all human activity, Newell's levels of human action. This is the idea that you can consider different time scales in terms of orders of 10, right? So 10 million seconds, that's months. A million seconds, that's weeks. And 100,000 seconds, that's kind of like days. That all lives in this, what are we doing? These are long-term planning. You might see theories from social phenomena. You keep going deeper, right? 10,000 seconds, that's on the order of an hour. 10 minutes, 1 minute. These are where like everyday tasks start to fit in. So the rational band, what's my plan for the day? What am I solving? How am I approaching things? How am I, making sense of the world around me moment to moment? But even deeper, we might go into individual actions on the order of 10 seconds, or cognitive operations, remembering a thing, committing a thing to memory, pressing a key even further, so on the order of 100 milliseconds. That's muscle movements. You don't get much faster than 100 milliseconds for programming a single muscle movement. This is this cognitive band, this idea of measuring things at this kind of more interactive level of when actual cognitive machinery is kicking in and doing its job, right? If I could only measure brain activity over the course of 10 minutes or an hour, I would only ever have access to things like the rational band, the everyday task type stuff. What's your task performance? Oh, your brain had elevated activity during that task. That's still good to know that, to be able to determine that. But getting deeper, to get deeper, we have to measure even finer grained. So, further down, on the order of 10 milliseconds, 1 millisecond, 100 microseconds, or no, wait, is that right? Yeah, microseconds. Now we're getting into what things like neural circuits do, how neurons fire, how the organelles within the cell work. And this is what you might throw out as the biological band. This is like, hey, we're like below human cognition. Cognition takes tens of thousands of these operations to make happen. So, I'll say ERPs kind of fit in right about here. We're getting measurements of the responses of collections of neurons, but we're hoping to tie them up further to say, this response happens in the brain when X event typically happens. What does that tell us about the rest of cognition? That's why people study ERPs in the first place. So, another aside. We're just kind of wandering into different little alcoves, but I hope these are clarifying and bringing into further higher resolution your understanding of what we're talking about when we're talking about measurement. We're trying to capture different phenomena at different levels. We're trying to meet them where they're at, right? I may not need a full 3D brain scan to get these ERPs. I could benefit from it, but I don't necessarily need it. What I need is the time. So, okay. Now, these are all cool. These are all cool individual things that we're starting to see, but we also need to talk about the real curiosity of the brain is not just how an individual thing works or where activity might be at any given moment, but more about what is connected in the brain. And there are two ways to look at this, and they're familiar. Structural connectivity, so are areas physically related? Are they touching each other, right? Are they wired up together? Versus how are they functionally connected? So just because something is wired up together doesn't mean they're necessarily always going to be active doing the same tasks and achieving the same things. So structural connectivity is the physical structure. So, how do the anatomical connections between neural elements fit together? So, let's give my honking voice a break, and we'll watch a little video about structural connectivity.
Trying to introduce the study of the brain to a bunch of students. I said, if understanding everything you needed to know about the brain is a mile, I asked the students, how far had we walked in this mile? I got answers, 3/4 of a mile, half a mile, someone said 1/4 of a mile. And I said, I think about 3 inches. This is unconventional science. There's just this huge gap in our knowledge about how brains work, largely because we have no idea what they actually are made up of at the finest level. When it comes to the nervous system, there are a large number of diseases where the only real sign that there's something wrong is the outward manifestation of the disease. A person is acting crazy, or they don't seem to learn very well, or their movements are disordered in some way. But if you look at their brain, with most of the techniques we have, there's nothing to see. You've got to see the wires. You just have to see them, and you have to see where they come from, where they go, what they connect with, and be able to map that out in enough detail that ultimately you will be able to render the information that goes into a brain to see the wiring diagram at the resolution of every single synaptic vesicle in every synapse. Enough resolution, we think, to see everything in a wiring diagram. I'm a lab head of a group of people who work together. It's kind of a shifting group of people, but it's difficult work and it requires many kinds of experts. So there are lots of people working together on this project.
What we're trying to understand is how cells in the brain communicate with each other. I have to look at the entire pipeline from the beginning, where we start with a mouse, make a sample, process the tissue, cut sections, and then ultimately begin imaging them. A slice is like you're cutting a loaf of bread, except you're trying to slice very thin pieces of tissue. So for a millimeter depth, you get about 33,000 sections.
Each section of the brain is next to the section that was sliced just before it, and the slice right after it is the next frame. And if you play these frames in sequence, you see the brain not over time, but over space, as if you're looking deeper and deeper into the brain. The big objects you see that appear and then slowly go out of appearance, those are nerve cells that are being cut through. And coming off of these cells are these large, light-colored objects, which are the dendrites. Every little object in here is a little wire. To keep track of these cells from one section to another, what you'd like to do is kind of color them in. So every one of these colors have no meaning other than to keep track of each object from section to section. What this allows us to do is to generate a wiring diagram based on this. We now know everything about these wires here, and from that we can generate all the connections of every cell in that area. Information forces you into this uncomfortable position where you have to kind of say, okay, I don't get it, but I know that the real world is more complicated than the way I'm thinking about it. I feel it's a very long road, and we've just started. That's my view.
So, what I hope that shows you is that there's a lot of connectivity in the physical structures of the brain. I can talk all day, say, the parts of the brain should be connected, but I hope this illustrates for you what that means. It is very connected. Things are very tightly blobbed together in there, and there is a intricate network of wires, so to speak, that run all through your head, all through your brain. It's all wires, baby. Your big wet computer model of how we think about a brain, right? So it's tricky to do, but it's important because we need to understand what is even possible. In the same way that doing a structural scan of your brain in MRI and then doing a functional scan as well helps us to figure out together, which parts of your brain were active when. This is sort of digging deeper to say, well, what's even possible? Like, these neurons can't affect these neurons directly because they don't have any connections whatsoever. So, structural connectivity it's multi-scale still. Because you can have physical connections between neurons that are, individual, so it's a single synapse between two neurons, or they can be entire tracts. The from early vision up through complex vision, up through all the spatial areas of processing. Hey, what does that chair look like? That kind of thing. And on most of the time scales we're talking about for, say, measuring activity of the brain, seconds and minutes, behavioral or smaller, these connections between the structural connections are going to be pretty static. They can be dynamic on longer time scales. It's like when you're growing up, your neurons will change, of course. But once you're kind of out there in the world, you don't expect a lot of change between the structural areas of the brain. It's just mostly set, right? Also, the connections between things can be binary. So just like these things are connected, or we can say, oh, these things are more or less connected. And that all depends on what tools we're using and techniques and all these things. In that last one, in the video we just watched, that's a binary approach. That's saying these objects, these slices, just color them. This thing is either the same thing or it's a different thing. And that's all we're doing right now, right? But further efforts might say, okay, how much is this neuron connected to this other neuron? So now functional connectivity. And this is all about, we've been talking about how measuring function is critical. We need to be able to track what happens in the brain over time to be able to understand what's going on. But this also means we need to be able to figure out, well, if this area is active and then that triggers this action in this area, seeing a flow, right? So which areas, what parts of the brain are connected? Now you see why we need both temporal resolution and some kind of spatial resolution, because we need to see, oh, this brain area seems to trigger activity in this brain area. Ooh, and that happens all the time. That's a track. Those are functionally connected. So anyway, the functional connectivity in the brain is all about patterns of connected synchronous. So they can either be one after the other or a simultaneous activity occurring over spatially remote areas, but also things that could be next to each other. But the point is that something that happens in one part of the brain and triggers an action in a whole different part of the brain, they're not especially spatially connected. They're not structurally very connected, but they're functionally very connected, right? So most tools for quantifying all this neural function produce these time series, and you separate them over the different spatial areas if you can, like our friend the fMRI. But those areas that are active, they're not always defined by a very specific kind of anatomical structure. It's all about the function. If this area says, ooh, I need to send this off to lexical processing, that doesn't mean that this area is necessarily structurally connected to it. That means that it knows there's a function that needs to be carried out. Careful, I always say I anthropomorphize these things. They don't know anything. I just send information where needed. But so this is all stuff that it's still a little in its infancy. We're still mostly measuring this by which parts of the brain are active together. So correlation being a strong statistical measure still. So which areas light up together? They must be functionally related. Perhaps not must be, but they could be. And that's where we start to ask the questions. There's a sort of a balancing point between these two. So the idea that functional areas don't necessarily need to be physically related, but physical relatedness does help to understand how things could be functionally connected, right? So effective connectivity is a term. It's the idea that try to refer to descriptions of connectivity that are actually causal in nature. So, right, we're trying to watch how this activity plays out over time and use our knowledge of the structure of the brain, our growing knowledge of the structure of the brain, what is possible. Pardon. Where are the highways, right? If this seemingly unrelated area needs to get information to this seemingly functionally related area. So seemingly structurally unrelated area needs to give information to a functionally related area. What is that pathway? How does that work? How does it get it there and not to other places, right? So using the knowledge about structure to make predictions about function, about what is possible. Models are limited in this field. Effective connectivity it's a growing field. It's a growing concept because more work like what we saw in the video is going on to map out what the structures actually are at this neurological level. So, great stuff, cool stuff. Developing field. That's what you're going to get for the rest of your life in cognitive science. We are not solved, buddy. So, structure versus function. They're never truly independent, right? Structure exists to support function, and function is constrained by the structure. Think about various animals, right? They all have similar structures. They all have things like, say, eyes, right? They all need the function of eyes, but their eyes work a little differently. An octopus lives underwater. A bunny's eyes are on the sides of its head, whereas a cat's eyes are on the front of its head. A bee has this complex mishmash of vision, like we've seen in their orb eyeballs. But the idea is that to understand each one of these eyes, even though they all have similar functions in the world, you need to understand their structure to make sense of them. So, this is a key idea. Coarser scales emerge from the interactions and activity of finer scales. So whether that's temporal or spatial or whatever. So for example, communication between neurons is fundamentally constrained by the physical properties of the axons of those neurons, right? Like literally down to the length and the width and the myelination and blah, 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 things we'll talk about in the future, don't worry. But this is the idea that the communication it's constrained by the structure, right? I can't communicate with something I'm not actually attached to in any way. So there's no single approach is going to really capture all of how all of the networks of the brain work. A physiological description, say a really good high-res image of a neuron, that's not going to include the temporal response patterns. Those are different approaches. And those differences of approaches reflect the constraints on the technology or the theory, right? So if I could have an imaging machine that could take electron microscope level detailed images of a very specific kind of neural, activity, like a bold response, and I could get it a million times a second, heck yeah, we'd be there, right? But we're not. That's not where the tech is at. We have to have these trade-offs. We have to come at it from different angles. So in practice, these approaches tend to actually come off as exclusive. An individual study of brain function isn't going to include tons of details on structural findings. And vice versa. That's just the nature of the way the research goes at the current state. But as I've illustrated is always the case, the tech will change, right? The tech will advance. We will get stronger imaging techniques. That's just going to happen. That's the way that our tech has been moving. And hopefully we'll start to see the convergence of results from different approaches, different studies to how we model what's happening in the brain. And we'll be able to connect those. Hey, connectivity at all levels, the interdisciplinarity, right? We'll be able to use better ground truth. Now, better empirical evidence, perhaps, is a better way to put this, from our cool tech to really tie together all these results to say, okay, what is connecting? Which things do fit together? And hopefully that starts to explain better the connectivity of the human brain. So that concludes our crash course on all things neuroimaging and the different techniques available. What are the differences between them? You should now be able to understand temporal versus spatial resolution. You should be able to understand what we're doing when we're measuring structure versus function. All these very good things. And we've come a long way. We've got away from the old way of just saying, hey, I'll measure the outside of your scalp to tell if you're a criminal. That sounds fun. And we've gotten into more complex things. And thankfully, there's a little bit more beauty to that approach as well. So, final thought. Consider how measurement resolution factors into things beyond medicine and neuroscience. How might a more temporally and spatially complete picture of our oceans, weather patterns, or astrophysics change things? Conversely, what might be the consequences of companies having exclusive access to our incredibly detailed internet activity, social media, and individual spending data? All right. Thanks. Take care.